Hello, guys. Welcome to another very interesting topic in biology with your very own Dinu sir. Okay, let's understand and enjoy this topic. Today, I'll be talking about a very interesting concept in genetics, which is called as transcription. What is the name again? Transcription. What is transcription? Remember the central dogma of molecular biology? DNA gives you messenger RNA, and messenger RNA gives you protein. Today, we'll be talking about the process of transcription. And transcription is what? First half of the central dogma of molecular biology, which means transcription is taking information on DNA and producing messenger RNA. Okay? Remember, very important, DNA is a double-stranded molecule, but the mRNA is going to be a single-stranded molecule. Is that clear? This process is called transcription. Now remember, we had used an enzyme during DNA replication, which was called as DNA-dependent DNA polymerase. What did that enzyme do? It took the information of DNA and produced DNA from it. Similarly, from DNA, I am producing mRNA. So the enzyme used for this process is called as DNA-dependent RNA polymerase. Are you understanding, everyone? Fantastic. Why it is called DNA-dependent RNA polymerase? Because you're taking the information from DNA and you're making messenger RNA. Am I clear? Fantastic. Let's understand a little bit about this process. Okay? Okay. Pay attention. I just told you DNA is a double-stranded molecule. Is that correct? I'm going to label this as dsDNA. So remember, the two strands of DNA are anti-parallel. Means they have opposite polarity. One strand runs from 3 prime, 5 prime, while the other strand runs from 5 prime to 3 prime. Is that clear? Fantastic. Now, from this double-stranded DNA, we have to produce RNA, correct? This RNA is going to be produced from only one strand of DNA. Is that clear? I repeat, RNA is produced from only one strand of DNA. Is that clear? And that strand, which I'm going to be using, make RNA from, this strand is also called as template strand. What is the name again? It is called as template strand. Is that clear? I repeat, strand of DNA from which I will produce RNA is called template strand. So what will be the other strand called as? The other strand will be called as non-template strand. Is that clear? Fantastic. Okay. So template strand is used to make RNA. Non-template strand is the other strand from which RNA is not made. Am I clear? Now, listen carefully. The orientation of template strand is going to be from 3 prime, 5 prime direction. You know why? Simple. Because mRNA will be produced from 5 prime to 3 prime direction. Again, in anti-parallel fashion. Why? Because the key enzyme produce RNA is called as DNA-dependent RNA polymerase. And RNA polymerase can polymerize and produce RNA only from 5 prime to 3 prime direction. Am I clear? Fabulous. So understand this concept. Okay, I'm moving to another slide right now to understand further. So listen carefully. I just told you that two strands of DNA. One strand is called template strand. The strand is called non-template strand. Is that clear? Which strand do I use to make RNA from? I make RNA from the template strand. Very nice. Now, RNA is always produced from 5' to 3' direction. The template strand should be oriented from which to which direction? Template will be oriented from 3' to 5' direction. And the non-template strand, because it is complementary, its orientation will be same as the mRNA, which is 5' to 3'. Is it clear? Now listen to one more very interesting concept. Now what happens here? Your DNA template strand will have bases. Which bases? Adenine, thymine, guanine, and cytosine. Let's repeat. A, B, D, C. A, B, D, C. Am I clear? What will be the bases in the non-template strand? Remember, they're going to be complementary. That means if there is a A in the template strand, there will be a T in the non-template strand. If there's a T in the template strand, there will be a A. If there's a G, there will be a C. If there's a C, there will be a G. Let's complete the non-template strand. E, A, C, D. E, A, C, D. Is it clear? Fantastic. I repeat, the template and non-template strands are the two strands of double-stranded DNA. They are not only anti-parallel, but also complementary. Is that clear? Everybody with me? Fantastic. Okay. Now what about the RNA? RNA, which is made from the template strand, also has complementary bases that of the template DNA, which means there is a A in the template, the RNA going to have U, Russell, because it is RNA. RNA do not have thymine. In RNA, thymine is replaced by uracil. For T, there will be a A. For G, there will be a C. For C, there will be a G. Continuing on, U, A, C, G, U, A, C, G. Do you see that? Remember, mRNA produced from template DNA strand in 5' to 3' direction, as well as in complementary fashion. Now notice something very interesting here. Look at the sequence of base of the mRNA. U, A, C, G, U, A, C, G. Look at the sequence of the non-template strand. T, A, C, G, T, A, C, G. Isn't it interesting to see that they both are same? Fantastic. So non-template strand and mRNA strand, because they are both complementary to the template DNA strand, their sequence of bases is going to be exactly same. Only difference will be in the DNA, it is going to be T, whereas same base in the RNA will be U. Correct? Therefore, the non-template strand is also called as coding strand. Why it is also called coding strand? Because basis of the non-template strand sequence is same as that of mRNA. That is why the non-template strand is also called as coding strand. It is an alternate name I have given the coding strand. The coding strand is also called as sense strand because it makes same sense as the mRNA. Same sense meaning sequence of bases is same. So if the coding strand is called as sense strand, can you tell me what will be the template strand called as? The template strand will be called as antisense strand. Is that clear? Fantastic. So remember, RNA is produced from 5' to 3' direction. The presence of an enzyme called RNA polymerase, in short RNAP, from the template or antisense strand, whereas the non-template strand, because it has the same base sequence as that of mRNA, is also called as coding strand or sense strand. Am I clear? This is the concept of transcription. Thank you so much. I hope you found it easy to understand as well as interesting. Thank you so much. I will see you soon with another very interesting and fantastic concept soon. Take care, good luck, and bye.